Hi, I'm Tori Clark and I'm a postdoctoral research scientist at the Australian National University, and in my research I'm looking at ways to improve photosynthesis so that we can grow more food to feed the world. And today I'm going to talk a little bit about how plants specialise the cells that they have so that they can become plants that can do photosynthesis and grow in different environments. So plants start from an embryo, which is inside a seed, and this is a single cell. And within that single cell we already start to see some differentiation. So the different ends of the cell start specialising and growing in different ways, so that as that cell divides we get a differentiation in the tissues. And in plants, two main tissue types are derived from that initial cell, and we call these meristems in plants. And these are kind of similar to stem cells in animals. So a meristem is a region of cells which can then further differentiate into lots of different cell types. And plants are quite interesting because they're a little bit different to animals. In animals, a stem cell is quite determinate. So if you want to become a heart cell, you would start with a stem cell of a heart, and that stem cell can then only produce functional heart cells. Conversely, if you wanted to be a liver cell, you would start with a liver stem cell. But in plants, they actually have this great flexibility in the cell types they can produce from their meristems. So from the shoot meristem, they can produce a range of different tissue types that produce the above ground parts of plants. So these are the stems and the leaves and even the reproductive organs such as flowers. And from the root meristem, it can produce all of the root tissue that's below the ground. So how does a plant cell know what sort of cell to become if there's such a variation in what's going on? And this is another way that plant cells are quite different to animal cells. Plant cells are really responsive to the environment around them. So it's about actually the location of those cells and what's going on around, what environmental signals they're getting, from the atmosphere, sunlight, gases, but also signals from the neighbouring cells around them. And those signals actually switch on different gene expression patterns within the cells. So you can think of gene expression patterns as basically like turning on and off functions within the cell. So if a plant was growing and it needed some structural support, it would want to produce cells that can be really strong. And so one of the ways plants do this is to have cells that produce a secondary cell wall for extra strength. And so the genes that control that would get switched on in those cells, and the signals would get integrated from the environment around it, so we would know that it needs to produce those different signals. So from a single cell in an embryo, plants can then differentiate right through to produce all these different cell types, which then go on to create the different tissues and organs that make the functional plant.